Why are you the way that you are?
The question I'm going to talk to you about today is so basic that we sometimes forget to address it completely. Why are you the way that you are? Why do you have brown hair and not black? Why are you tall and not short? Why am I stunningly good looking, whereas the person behind the camera is less so? Well, we don't exactly know the answer to all of these questions, but we've got an idea, and we've made some major strides in recent years, particularly in a field called epigenetics. And before I explain what epigenetics is, let me present you with 3 cases. The first case involves 2 identical twins. Identical twins contain exactly the same set of DNA. We can learn a lot about our question of the day by seeing where identical twins differ, particularly in cases where one twin gets a disease and another does not. The rate at which this happens is called a concordance rate. In identical twins, the concordance rate of achondroplasia, a rare type of dwarfism, is 100%. If one twin has achondroplasia, the other has it, every time. Concordance rates for other diseases are much lower, though. For example, the concordance rate of schizophrenia is only 50%. If one twin has schizophrenia, the other only has it 50% of the time. The concordance rate for breast cancer is just 11%. My question for this first case is simple. If we are dealing with genetically identical people, how can this happen? If their genes are identical, why don't they both get the disease? Case number 2. An adopted infant is brought up in an abusive family. He is neglected, shown very little love, receives poor nutrition, a poor education. As the child gets older, let's say 10 years old, we move him from one family to another one. This new family is very loving, very caring, yet regardless of a change in environment or counseling, the boy has major emotional difficulties. Even though we've changed his environment completely, for the rest of his life, the boy will be prone to anxiety and depression, likely to use drugs, has a lower life expectancy. My question for this case is, why are these events in early childhood seemingly life-lasting in their effects, even after the memory of these events has faded? And the final case looks at neurons. Every cell in my body has all the genetic material needed to create my entire body, including my neurons. And why aren't my neurons producing digestive enzymes, for example? Why aren't my neurons producing hemoglobin? The answer which science has provided to these questions has been twofold. First, the genetic code you were born with, and second, the environment in which you have lived. Or put a little more sexily, nature combined with nurture. For those who haven't heard this before, nature refers to DNA, the unchanging genetic code you were born with, which is simply a really long string of bases that is present in every cell in your body. There are only 4 types of bases that make up DNA, and any string of these bases is referred to as a gene. Genes code for every single part of your body, your eye color, the shape of your nose, the way your brain is arranged. And on the other side of the coin is nurture, the environment we are in, which is constantly affecting our genes by switching some of them on and some of them off, and through this interaction between nature and nurture, we get a human being. Well, none of this nature versus nurture stuff is wrong, but there is a major problem with it. Primarily, it is extremely vague. There has to be a biological explanation for what is happening between nature and nurture. Something to tell us how our environment is taking this blueprint of DNA and doing different things with it depending on the situation. Epigenetics is this biological explanation. Epigenetics refers to the short-term, generally reversible modifications to DNA that cause it to be read in different ways. And we're starting to understand these changes in great detail. Let me start with a basic example. Let's take a look at this DNA base, cytosine. There it is in its naked form. If I temporarily attach a methyl group to this cytosine, I've made an epigenetic modification. This one is called methylation, and the environment is making modifications like this one to our DNA every single day. Why would anyone care about this? Well, if I methylate a whole gene, in other words, if I attach a methyl group to each base pair in a set of base pairs, I silence the gene. I turn it off. Well, it turns out that the hemoglobin gene in my neurons is very highly methylated, and therefore, it is turned off. It gets better. I can attach different things to these genes. If I attach an acetyl group to the individual base pairs of a certain gene, I can turn that gene on. I upregulate. Now imagine the implications of being able to turn genes on and off at will. We could turn unwanted genes off. We could turn desired genes on. And we can completely unlock the potential of the human genome. So back to the case of the twins, particularly the case where one twin has a disease and the other does not. We found an epigenetic reason for this. Recent research has found abnormal rates of methylation in many mental illnesses and almost every single cancer we know about. In cancer patients, parts of the genome are too highly methylated, specifically the genes that code for tumor suppressor cells. This methylation diminishes our defenses, allowing the cancer to grow. If we could go into the genome and remove these methyl tags, we could put up a fight against the cancer. And it turns out there are currently 2 FDA-approved cancer drugs that do exactly that. Additionally, if we could figure out exactly what environmental factors caused this methylation in the first place, we could avoid it completely. In reality, epigenetics is a bit more complicated than methylation off, acetylation on. A man named David Alice at Rockefeller University has shown that there are at least 50 different types of epigenetic modifications in addition to methylation and acetylation, and these don't exactly function as an on-off switch, but as more of a volume dial. Genes get turned on and off only to a certain extent. They're shades of gray. On top of that, not only is DNA modified epigenetically, so are the special proteins that DNA is wrapped around, called histone proteins. This can have effects on how long these modifications last. Our epigenetic modifications during childhood, such as the ones caused by abuse and neglect in case number 2, seem to be almost impossible to reverse on a biochemical level, and we can see this manifested behaviorally. So epigenetics is a complex field, and it looks like we are only scratching the surface. There will undoubtedly be many opportunities for future scientists to study this integral biological link between nature and nurture, so that future generations can have a better answer to the question, why are you the way that you are?